Hello, and welcome back to the Biology 219 video playlist. Last time we talked about the genetic code and how cells read it. Let's get into the details of that process. The first step to making a protein is actually making something else, RNA. Cells use transcription to make RNA intermediates. This is useful because the same code can be read many times to make many copies that can be used to make many more proteins. The process of making RNA is similar to all other macromolecular synthesis. Remember that RNA and DNA are really similar molecules. Their structures are nearly identical, except that RNA has an extra hydroxyl group at the 2' carbon. This hydroxyl group makes it hard for the bases to hydrogen bond with other nucleotides, so RNA is typically single-stranded. RNA monomers are already high energy, so they don't have to be attached to a carrier molecule. They are connected together through condensation reactions carried out by the assembler RNA polymerase. The act of making RNA takes place in 4 steps. The first step is recruiting RNA polymerase to the right part of the chromosome. RNA polymerase doesn't have any specificity. The scientific term is promiscuous because it will copy any region of DNA. It is targeted to the right location by 2 kinds of factors. Some of these factors are built right into the same DNA, and these are called cis factors. Other factors bind to cis factors. These are usually proteins that were built in an other location, so these are called trans factors. We'll talk a lot about these in class. Once RNA polymerase has found the right spot, the second step is initiation. This is opening the DNA and beginning to read it. The third step is elongation, where the new RNA polymer is elongated through a series of condensation reactions. Remember that the order of RNA nucleotides is determined by the DNA. RNA polymerase uses the same AT and CG base pairing rules that DNA replication does, although in this case, RNA polymerase puts U's instead of T's. The last step is termination. This is a complicated process, and we'll get into the details in class. For now, it's enough to know that RNA polymerase can't keep copying forever, and there must be a signal for it to stop and for the RNA to be completed. Finally, transcription is used to make lots of different kinds of RNA, including mRNA, tRNA, rRNA, small RNAs like microRNAs and small interfering RNAs, and new types that have just recently been discovered. These RNAs are defined by their function and not their structure. They are all chains of RNA nucleotides connected by covalent bonds. RNAs are often neglected because they are temporary and their main function is in building other functional molecules. But many RNAs have functions like splicing RNAs, signaling RNA production and degradation, and regulating gene expression. In class, we will see how these RNAs are built and processed and read to form proteins.